I think when you think about the situation when a diagnosis of inherited retinal degeneration is established, and by the way, from now on, I will not say inherited retinal degeneration, I will just say IRD for short. So once a diagnosis of IRD is established, the next question that comes up is what can be done? And it is my pleasure to share with you this evening, some aspects of the developing therapeutic options and directions. So as you all know, IRDs are actually a group of diseases which can be very different from one another, but all of them are characterized by the progressive loss of the photoreceptors and the cells in the retina, including the retinal pigment epithelial cells, or RPE, which basically lead to vision loss. Now, until now, IRDs have been considered incurable, but we're very fortunate to be witnessing the beginning of a historic shift in this regard, because with the recent rise in research efforts in relevant fields all over the world, several treatment options for IRDs are now becoming available on the horizon. Now, largely speaking, the leading therapeutic directions for IRDs include mainly gene and cell therapy. And the eye is an organ which is very suitable for such therapy. First, it is small and can be easily accessed. It needs a relatively small volume of the injected material is required. Typically, an immune response to the genetic or cellular materials which are being injected is relatively limited, and so is the systemic spread from the eye, which is an important safety factor. And finally, advanced imaging systems of the retina, which are very commonly available, enable us to monitor the response to treatment in a non-invasive manner. And indeed, over recent years, there has been a significant surge in research of new genetic therapies for IRDs, with an impressive rise in the number of participating subjects and centers all over the world. Now, when we speak of genetic therapy, it's interesting to note that there are several possible approaches. Classic gene therapy, which is also called gene replacement or gene augmentation, basically means insertion of a functional copy of the mutated gene to the target cells. And in contrast, genome editing using novel approach called CRISPR is a different approach which enables to edit the human genome within the affected cells, in this case in the retina. Now, in either case, gene therapy requires a vector for the transfer of the genetic material into the target cells. And in the case of retina, adeno-associated virus, or AAV for short, is considered the safest and most effective viral vector platform, at least to date. And there are certainly clinical trials for several genes which are involved in different IRDs, which are presently underway. And on this slide on the right, I've listed a few of the conditions, including Leber congenital amaurosis, choroideremia, retinitis pigmentosa, Usher syndrome, and achromatopsia with the involved genes, which are listed as well. And of course, we cannot go without mentioning RPE65, which is a gene which encodes a protein which plays a key role in the visual cycle and is therefore very important for the entire visual process. Now, defective RPE65 gene leads to an early onset retinitis pigmentosa and Leber congenital amaurosis. And as you all know, in December 2017, the FDA approved the first gene therapy, which is called Luxturna by Spark Therapeutics, to treat subjects with RPE65-related or associated retinal dystrophy in the US, and the situation in Israel is following. The drug is now also approved in our country, and so far 11 patients were treated, mostly at the Hadassah Eye Hospital in Jerusalem, and I think at least some of these subjects were treated directly thanks to the efforts of the Israeli consortium, and for us it was very exciting and almost thrilling to learn about this privilege to treat patients. Another unique approach for genetic therapy, which was developed in Israel, actually was developed by Professor Dror Sharon and Professor Eyal Banin, whom you'll meet in a short while, together with researchers from Bar Ilan University, employs RNA editing. Now, this method uses enzymes which are self-occurring to edit mutated RNA, causing a variety of IRD phenotypes. Now, unlike DNA editing, including CRISPR method, which I've just mentioned, RNA editing can be fine-tuned and is less risky. And it is my pleasure to share that the group showed initial success in vitro studies, and therefore in vivo studies in the mouse photoreceptors are presently underway. So hopefully there is a lot of exciting news that are coming up ahead. A different approach for the treatment of IRD is cell replacement therapy, which aims at replacing damaged host cells with transplanted donor cells. Now, for IRDs, this direction has mainly focused up until now on transplantation of the RPE cells, because these cells, as I said, support the photoreceptors and are vital for the retinal function. And in fact, the RPE cells are important for the entire visual process. Now, quite a few different mutations which affect the functions of the RPE can cause IRDs. And one advantage of cell therapy is that this is a mutation-independent approach, which can therefore work even when the cells are completely lost, because the lost cells are simply replaced by the transplanted ones. There are several options for the types of cells which can be used as the source for the transplantation. And the main use, at least in the present time, is of induced pluripotent stem cells, or iPS cells for short, which are becoming a very attractive strategy for IRDs. Now, very briefly, iPS are cells which are generated from mature body cells like the skin fibroblasts or peripheral blood cells, which are collected from the subject and are reprogrammed in the lab to become stem cells. The derived stem cells can be then differentiated again in the lab into various cells which are used for the transplantation, including, for example, the RPE. Now, this approach faces several challenges in the present time, but has a promising potential for IRDs. Now, the transplantation itself can be done either by injection of the solution under the retina, which basically requires a surgical approach, which is done in the operating room, or via intravitreal injection, which theoretically can be done in the office. Each option carries different advantages and disadvantages. And again, it's exciting to share that there are several ongoing phase 1 or 2 clinical trials, which are looking at the efficacy of stem cell therapies for IRDs. And in this regard, it's my pleasure to mention the new Israeli Consortium for Innovative Stem Cell Research for Clinical Use, which was recently established, and which aims to bring together leading scientists and pioneer physicians with a goal to accelerate the development of stem cells for transplantation in various human diseases. And of course, the RPE and retina are included and listed as a top priority. Now, in the future, it seems that a combined genetic cellular approach will become important for the treatment of IRDs. Because if we think about it, if we think about iPS, for example, it's important to note that because these cells are derived from the patient, the cells carry the same genetic risk factors that the patient suffers from, and therefore they need further genetic manipulation to correct the mutation within them and be healthier. And gene editing technologies such as CRISPR and their combination with the development of the iPS cells has the potential to repair patient-specific mutations and opens a new era of personalized precision medicine for our patients. So to summarize, it seems that both the genetic and the cellular approaches hold a very great potential to become successful treatment strategies for IRD patients in the following decades. Now, some of what I say may sound imaginary, but is already happening right now much thanks to the deep commitment and combined efforts of everyone involved, including you. So indeed, the future is bright. And with this, I thank you for your attention.